Hello. All right, let's try this with subtitles. All right, so here is chapter 10, and I'm going to go through the highlights of it. There's definitely more in the book. By all means, have a look at that. So, the first thing that comes up for this particular section, and we're only doing parts of chapters 10 and 12 in this one, the first thing that comes up is this table. And so, have a look at it. This is one that I would say it's good to get a general sense from this of exactly what's going on. So let's look at each kind of antibody in turn and see if we can figure out what it does for the most part. So they're up here very helpfully in kind of the order in which they appear in the genome. So I think that helps a little bit. And the order in which they would be produced, more or less. The E comes, I believe, before the A in humans. So, IgM, first antibody produced. It's great. And remember, IgM's partner is IgD. So if you make some IgM, you're going to get some IgD as change. It's like those little pennies that you get after you buy whatever. What are they for? I don't know. They just make up the transaction. So you can see IgM is not great at neutralizing. So neutralization means stopping a viral or bacterial infection before it starts. It's basically the thing we talked about with a snowball. Like if you're completely covering over the outside of the virus, then there is literally no way that they can grab onto a cell or do anything. You're paralyzing it by coating it in antibodies. And so IgM can do that, but it's not the best at that. There basically aren't very high IgM levels ever. Opsonization, it's okay. And so this would be a pathway that would eventually lead to complement-mediated lysis. In activating the complement system, it's pretty good, and we'll show you why here in a couple of slides. But basically, do you remember the C1q protein? It kind of looks like, I don't know, it looks like one of those massagers with all the little prongs on it that you can use to, they feel nice on your head anyway. That protein, C1q, needs to be holding on to multiple antibody FCs in order to get binding and start the complement activation process. And IgM happens to have 5 FCs right next to each other, and they can't get apart from each other. So that's why it's good at that particular thing. And I'll show you pictures in a little bit. There's nice electron microscope pictures and drawings. Transport across the epithelium, you actually do get a little bit of IgM in stuff like breast milk. But for the most part, it's not going to do anything for your NK cells or your mast cells, etc. And you can see from the mean serum level, it's not high. And generally, these are all your new, sort of newly minted B cells that are just trying out their brand new antibody. And a lot of them, frankly, aren't going to make it past the IgM stage, but they did great to get this far, so fine. Now, with your IgM, you're going to get IgD, and so IgD is another interesting one. It's a version of the antibody that is probably an evolutionary relic. It does not have many functions anymore, but the one function it's still got is that this is basically bling for your mast cells and your basophils. So cells like that will have receptors for IgD on the outside, and so they can actually decorate themselves with IgD. And if you've got IgD there, it's something that you've just made IgM against because they're made from the same messenger RNA, differential splicing. So the same pre-messenger RNA, rather. And so, yeah, it's going to be something that is useful and something that's current, something that's, very hip and now, just like just like clothes are supposed to be, right? Yeah. So this, by binding on to IgDs, these mast cells, basophils, etc., are not going to be sensitized. They're not going to be more likely to degranulate and cause all that, histamine, that sort of thing. But what they are going to do is they are going to be able to bind to additional antigens. So this is a different way to just stick some roving bacterium to a mast cell. And it's not going to go well for the bacterium in general. So that's how they help. But for the most part, we could probably get by without them because these cells would also have the ability to bind various IgG subclasses and IgE sometimes. So yeah, I don't know that we super duper need it anymore. But whatever, that's genetics. You get what you get. You don't get to pick. All right, IgGs, 4 different subclasses, slight differences between them, but let's just compact these all into one and say, all right, the average is roughly where IgG is going to be. So pretty good at neutralization, great. Pretty good at opsonization, fine. And in this case, the opsonization and activation of the complement system, this is going to be through binding multiple copies of IgG. So you've got to get 2 independent IgG hits to set off complement, whereas one IgM, that great big star-shaped thing binding, is going to be enough to set off the complement activation system. So, yeah. IgM is a more efficient way, but it's also going to be more error-prone. If it binds to the wrong thing, you're going to end up with complement activation where you don't necessarily want it. So, yeah. IgG, it's like 2-factor authentication for your immune system. Oh, how nice. Fine. Let's see. Other things that it's good at. So does it get transported across the epithelium? No, it does not. You generally won't find much IgG on mucosal membranes because it just doesn't have a good way to get there. And that's okay. IgG is for something else. You get a lot of it in the bloodstream. And it is the one that's produced in the highest levels. You can actually sum these together, 9, 3, 1, and 0.5, and that's the total IgG level. And since they're all kind of related and have overlapping functions, they do a real good job. They're the thing that keeps the bloodstream sterile, effectively. Transport across the placenta. So these are antibodies that can go from mother to baby via the bloodstream. Remember that when they're drinking the breast milk, all they are doing is coating their little throats and, I guess noses if they have like a, if you make the baby laugh or something and it spits milk out its nose. But yeah, pretty much you get a couple of mucosal membranes coated in antibody and very little else for your money. It helps, it definitely helps, but it's not this total protection that it would be nice if it was. Diffusion into extravascular sites. Now that's interesting. So this is IgG will sometimes leak out through the endothelia, like the vascular endothelium, and you'll find it in the sort of tissue space just outside of that. So IgG can have some effect outside the bloodstream, but it's basically going to be in the site that's close to the bloodstream with sort of active liquid transport of some version, from the bloodstream. So that's the idea. It's in the blood or close to the blood, and that's pretty much all it does. IgA can neutralize, and IgA is going to be your main neutralization weapon on mucosal membranes. It's the thing that stops problems before they become problems. IgG is more of a cleanup crew. If anything is in your bloodstream, something has gone horribly wrong, and IgG is there to try and patch it up, but, it may be rough for a while. The other thing that it's particularly good at, so IgA is not great at activating complement, and there is some in the bloodstream. So this is the serum level, so think of this as like the amount in the blood. But it tends to be the monomer that you find in the blood, and it'll be that monomer that can also leak out just outside the capillaries, veins, etc. And it works more or less like IgG in that case. It's going to hopefully neutralize any pathogens that are out there. The one thing where IgA absolutely excels is in transport across the epithelium, and as we will see, the IgA actually has to be hooked up back to back, so it's 2 IgA molecules, and they're chained together by a little thing called the J chain. J chain? Yeah, J chain, yeah, which joins them together. And there is a special transporter that can only transport joined up IgAs, and it'll transport them from the blood side of the membrane over onto the mucosal side of the membrane. So that's just how it works, yeah. And so that's how IgA gets all over your mucosal membranes. It is deliberately transported there, at cost of energy and time and everything else. So it's, we mean to do it. And then the last thing you got is IgE. So IgE is going to be an activator, like a sensitizer of these mast cells. IgE is going to be a fast way to provoke a sort of innate response where you're going to get the histamine, you're going to get the inflammation, these sorts of generic changes that make it a little harder for different kinds of pathogens to grow and can generally help the other components of the immune system. So IgE is there to kickstart this, and the big problem comes in that now that we've evolved all these other immunoglobulins, they basically take care of the necessary functions. And so you've got IgE there, which is great at setting off the sorts of cells that are involved in allergic reactions. And generally, it doesn't have a whole lot else to do. So IgE ends up, just because it's kind of lacking some other fundamental purpose, it ends up being the thing that's most associated with allergy. And you can see, thankfully, the level in the serum is real small, 10 to the minus 5. Yeah, that's very, very small compared to all these other ones. So you don't want a lot of IgE around ever because it's powerful, and you don't need a lot of it, and probably we have more IgE than we do need. So there we go. Yeah, and I summarized a little bit of that at the side. These are a new slide set, so I don't have all the definitions on there. So you're going to have to, in your notes, make some of your own definitions, and I think that's all right. Yeah, it's a good way to learn. All right, so now let's show some of the things we were just talking about, but with pictures. Isn't that better? I think it's better. So down here, you have this little thing called PIGR. And PIGR is a polymeric immunoglobulin receptor. So there are lots of immunoglobulin receptors in the world, like the things that bind IgE to a mast cell, for example. In this case, it is going to be specific for a polymer of IgA. And so it's going to have two that are back to back, and so, wouldn't you know it, that's how IgA is going to get bound. And then once this is bound, it's actually going to get pulled inside of a little vesicle. It's going to bypass going to the lysosome, which would be the normal order of operations. And it's instead going to get delivered from the bottom to the top of your nice columnar epithelial cells. And then once it's there, it'll be loose. And so, yeah, you'll have your IgA floating around there in the gut epithelium, for example. And if the IgA is specific for something, it can bind to toxins, it can bind to actual bacteria, it can bind to viruses, and it will generally do its best to keep you safe. Okay, so, yeah, the neonatal Fc receptor carries IgG across the placenta and prevents IgG excretion from the body. So IgG is in the blood because we work hard to keep it in the blood. We make a ton of it there, and there are actual blockades that don't let it out. That's basically the idea here. So here's the Fc part of the antibody over here. Here's the Fc receptor here, and you see it's got a different shape. And actually, from the look of this, it looks a lot like one of those display molecules. It looks like a MHC-related molecule, and I bet that's a beta-2M sitting there underneath it. But in this case, it's going to be an Fc receptor, and so great. And so the Fc is binding to this little molecule down here for some... Oh, no, those are glycans, sorry, on the Fc. And the Fc receptor is going to bind to the edge of it. So there is a special receptor for Fcs of IgG in the placenta. So the placenta is like this little thing that basically feeds the baby. And so this is the idea. You have a nice little baby here, and mom is making all kinds of antibodies. So while the baby is still inside of the mother, you can get, she'll make, all this nice dimeric IgA, that's on all the yellow zones, which is going to be basically all of her mucosal membranes. So think, like nose, throat, lungs, and basically entire intestinal tract. You can imagine it spiraling around behind there. I'm assuming that's meant to be small intestine and large intestine. Maybe that's like stomach and small intestine. I don't know. We're just schematic here. There'll be a little bit of IgG in the bloodstream and a little bit leaked out into the tissues, so they're basically highlighting all the rest of the body in that. IgM is going to be, they've got it shown here mainly in the heart. That's interesting. And a little bit of IgE as well around some of these areas, but relatively low expression. The IgG, though, gets into the placenta and into the uterus and into the literal baby because it is actively transported across there. So, all right, I think that's good enough for this particular thing, and then we will finish out this chapter in the next section.